is, I usually have a quiz on this stuff. The last quiz we did was in class. The next quiz for this one, I put online. And so you'll be able to take it on D2L. It'll be based on the pictures I showed you here today. And you have the study guide, so you know what you're responsible for. Since I'm doing it today, originally I wanted to make it available all the way through Monday of next week, so I'll stick to that. But I'll make it available today, since I'm presenting today. I don't know if anyone's going to take it today, I doubt it, but it doesn't matter, it's online. I'll just hit the button and there you go. So take your online slide is just cardiac muscle. I might ask you that. Identify tissue type, cardiac muscle. The only structure I might have you identify is intercalated disc. All the vertical dark bars, any of them, I can just point to one arrow, point to any of these little dark bars, intercalated disc. Notice all the cell nuclei. There's a lot of nucleus, but that's not the intercalated disc, it's the vertical bar. Remember, those function to kind of glue the heart muscle together. So just know that. It's cardiac muscle and intercalated disc. So the heart pumps blood into the arteries. Let's start with the largest class of arteries, the elastic artery. So I might ask you, identify a vessel type. And hopefully one of the choices is elastic artery. You can see the thickness of the vessel wall. You remember your tunics. On the big ones, how many layers do you have? Three. You have the innermost layer, TI. Okay, TM is basically all this stuff here. So here's TA. Tunica intima, media, and adventitia. Now, the tunica media, let's remember our tissue types. The tunica media has elastic tissue and smooth muscle. And the tunica adventitia is mostly loose connective tissue, basically. One other thing I usually have you identify as a structure is the vasovasorum. The large vessels are very big. They don't have access to nutrition from the blood that flows through them, so they have their own little vessels, shown here. We call that the vasovasorum, which is our study guide. Okay. One other thing about tunica intima that you can see on other pictures, say this one here. This is the artery and vein slide, and in terms of vessels, this is not an elastic artery. This is more a muscular artery. Okay, so as far as vessel type, that's what it is. This is, we call this large vein. Again, the large ones, both of these have how many layers? Three. Okay. So if we look at a close-up of the muscular artery, this, we see TI, tunica intima, tunica media, tunica adventitia. From about there to there is tunica adventitia, there to there is tunica media, and this is tunica intima. If I point to this dark squiggly line and I say identify structure, it's internal elastic lamina. It gives that tunica intima that rippled appearance. Because remember what arteries do, they kind of expand and collapse, and so that kind of implies that. And let's remember that it's smooth muscle and elastic fibers, and the TM, and mostly collagen fibers here. But if it's dark staining tissue, what kind of tissue is that? Elastic fibers. The TM has less elastic content in the muscular artery. It's mostly smooth muscle. You can compare that to the elastic artery. There's a much higher elastic content, right? That's why we call them elastic arteries. Here's the large vein, and you should be able to recognize our three layers. TI, from here to here, we're going to call that TM, tunica media, and from here to there is tunica adventitia. Yeah. So that's our vein. Is that artery or vein? I think it's an artery. Usually, how I tell, TM. Look at all the muscle content there. TI, TM, TA. Okay. This is an arteriole in the white pulp. is white pulp. Spleen. How many layers do arterioles have? It's not 3, it's 2. They have a Ti and a Tm. So if I point to the innermost layer, by default, what layer is it? Ti. So you see you have a little bit of Tm there. By definition, it's about 2 layers of smooth muscle, by definition, is an arteriole. So that's a pretty good arteriole for our class. 2 layers on the spleen. This is from rat testes. This is a muscular artery. So how many layers? 2 or 3? There's going to be 3. Ti, Tm, Ta. Okay? And those are red blood cells in the muscular artery. This is from the rat testes. You see little rat sperm right there. But we're going for the blood vessels here. This is also a good spot for capillaries. That's a capillary. It has some red blood cells inside there. 1 layer, just Ti. So if I say identify layer, there's only 1 layer to put. Tunica intima. Okay? And it's a capillary. That's the vessel type. That's all I can ask you for that. For an example of a small vein or venule, we have the central vein. This is from liver. I call it the shower drain, right in the middle. We'll talk about liver. That's central venule. Okay. Here's a closer up picture of the central venule from the liver. Just know that. Moving on to the blood. So this slide, obviously, I want you to identify these 2 cells. What's the small fragment? Platelet or thrombocyte. Monocyte, that's a pretty one. I probably got really happy when I found that one. They're tough to find. This slide is eosinophilia. You should have a lot of eosinophils, and I see 4. 1, 2, 3, 4. Eosinophilia is the blood condition. Here's a close-up of an eosinophil. It's a little blurry, but you can still see the red granules in the bilobe nucleus. That's the hallmark of eosinophil. Here is, these 2 arrows are pointing to the same cell type, neutrophil. That's all you need to know for that, neutrophil. Moving on. This blood condition is iron deficient anemia. That thing, microcyte. Okay? Basically, microcytes are small iron, low hemoglobin, low iron containing cells. And they kind of clump up like this because of the low crit levels in iron deficient anemia. This is sickle cell anemia. Usually when I see the puckered or sickled cells, like this one here or that one there, it's sickle cell anemia. This is pointing to 2 different cell types. The one on the left, lymphocyte. This one, you should be able to tell me. Red granules, bilobe, eosinophil. Okay, let's move on to trachea. We did this today. You should be able to identify the 3 layers of the wall: mucosa, submucosa, adventitia. If you look at a closer up view, we see the tissue type is ciliated, pseudostratified, columnar epithelial tissue with goblet cells. You should be able to name the tissue type. You should be able to identify that cell. Or that, what would you call that? Cilia. What would you call this? The lamina propria. This is the submucosa. The only structure you have to identify are the seromucous glands. Any of these, right? The cuboidal epithelium is your dead giveaway gland. This picture shows a little bit of hyaline cartilage. So what layer is that? The layer is adventitia. But what if I say, what's the tissue type? Not adventitia. Hyaline cartilage is the tissue type. Okay, that's part of those C-shaped rings. Okay, Let's move to lung tissue. All right, well, bronchiole. What's that? Hallway. Alveolar duct. We're going to point to just that. Alveolus. What about this? Alveolar sac. Okay. Here's a closer up view. This is, I might have you identify the tissue type. I describe it as a simple cuboidal ET with sparse cilia. It's of the bronchiole. Okay. So, again, same questions. Bronchiole leads to alveolar duct. That might be an alveolar sac. These little ones might be individual alveoli. That's all I'll ask you for healthy lung tissue. Emphysema lung tissue looks like this. Looks like a big mess, doesn't it? The thing about emphysema, it destroys the alveoli. So what it does is it takes all your small, perfect little alveoli, it obliterates them, so what you have are fewer, larger alveoli. And in this case, larger is not better, it's much worse. I mean, look at this. Does this look better? This is terrible. This is tissue that's not going to do you much good. So usually, the only thing I have in your study guide for emphysema lung is these larger alveoli, which totally, I mean, lack of a better term, they suck. I can do anything for you. Look at this. Emphysema is a horrible disease. It destroys the elastic fibers. It destroys the integrity of the alveoli. Emphysema lung, healthy lung, you can see a remarkable difference. Okay. That's it. That's what you're responsible for an online quiz. Fun time. I mean, is this class?
Yeah, I've seen.
Yeah. I was going to send it in the email link that everyone sent the last video.